become is not determined by genetics alone. Our environment, whether nurturing care or unpredictable stress, has biological imprints on our brain and behavior. My project explores this intersection of nature and nurture, investigating how early life adversity impacts the neurological circuits behind behavioral implications. Early life adversity, or ELA, refers to traumatic or stressful experiences that occur in early life, such as poverty, neglect, or abuse. Contrasting a nurturing upbringing, early life adversity increases risk for the development of psychiatric disorders in adulthood. As studying early life adversity in humans is limited, our lab has developed the limited bedding and nesting model to model early life adversity in rodents. Here, we simulate a resource-scarce environment to induce stress on mouse pups. Our early life adversity paradigm occurs at a critical period during early development and produces significant cognitive impairments and dysregulated reward behavior in adulthood. When we test our mice as adults, we find that early life adversity leads to dysregulated reward-motivated behavior. Our early life adversity mice have a depressive and hedonic phenotype with reduced interest in rewards, such as highly palatable food and the scent of an opposite-sex mouse. We then wanted to investigate the neurological mechanisms behind the behavioral impacts of early life adversity. Our lab tackled the question of where in the brain is early life adversity sensed and encoded. We used a genetic tagging technique to permanently label populations of neurons that are active during early life. We then quantified these tagged cells across a number of different brain regions and found that the only region that discriminated between our early life adversity and our control conditions was a region called the paraventricular nucleus of the thalamus, or the PVT. As you can see in the slides right here, early life adversity leads to increased activity in early life in the PVT compared to our control condition. We've also monitored real-time activity of the PVT in response to reward. In our control mice, we see a reduction in activity when reward consumption begins. Our early life adversity mice, however, seem to be unable to turn activity in this brain region off. Now that we know which region of the brain is responsible for our dysregulated phenotype, we want to manipulate this region in order to try to restore interest in reward. To do this, we were able to inhibit those early life neurons we saw a few slides back through the injection of a drug into the brain. By inhibiting these neurons, we find that we're able to prevent the depressive phenotype in our early life adversity mice, essentially bringing back their interest in palatable food. To sum everything up, early life adversity leads to anhedonia, or reduced pleasure, which is a core symptom of depression. Early life adversity also leads to increased activity levels in a region called the PVT, permanently altering this region. By manipulating these permanently altered neurons, we were able to prevent the depressive phenotype in our early life adversity mice, bringing back their interest in rewards. Our project is highly translatable to human experiences and emotions. By getting a better understanding of the neurological and behavioral implications of early life adversity, we know how to better address the cognitive, emotional, and behavioral impacts in those who experience it. Thank you for listening.